I am told to give a final exam. Okay, I will give a final exam. You are to give it, Dr. Abbott, during the final examination period. Yes, ma'am, I will do that. You follow me? That's what we're doing. And it's 50 minutes long, and it's 50 questions, and it's the same as every other exam you take in this semester. So please spread the word. All right? Those of you that have actually read the syllabus. And again, you know, this is sort of fascinating. Okay, so that's it. All right, what do we need to know? Well, we do need to know how an action potential is in fact generated. Yes, and it's not going to be enough to know that there's a depolarization piece, a repolarization piece, a hyperpolarization piece, and of course this threshold potential. You are also going to have to describe what's going on with regards to the nerve cell at these particular points. So, for example, is at this particular point, to start this, is sodium primarily moving into the axon, or is it moving out of the axon? Is potassium moving in? Is it moving out? What is happening with chlorine? Again, one of the things you need to understand, a cell, it's a beautiful thing. It has the ability to control whether ions leave the cell or not, because it has a phospholipid bilayer with a hydrophobic region. So it's quite easy for a cell membrane to regulate ion movement, not water, but ion movement. So it doesn't have to let chlorine go anywhere. It can keep it within the cell cytosol. It doesn't have to move it. And then it can simply move another charge. Remember, when we look at the generation, or in addition, when we look at the generation of an action potential ultimately becomes an electrical chemical signal, right? But remember that the actual signal itself, if we say it's electrical chemical in nature, the charges move not only because of a difference in charge. Sodium moves in. It's attracted to the anions, which are in the cytosol. They're being held there deliberately. So sodium gates open and allow, in other words, it's literally with like charges, but chlorine and negative proteins can't move, they can't get out. So sodium moves in, it's attracted, like charges are attracted. So that's one reason sodium moves in. But the other reason sodium moves in is because there's a concentration gradient that allows sodium to move in. In other words, there's more on the outside than inside. The cell has deliberately created that so that it can create a resting potential. So when we look at an action potential, we say, oh, how do I describe that? What in fact is it? Is it electricity? Is it, well, yeah, but it's also generated because of a difference in concentration. So we generate an action potential. We're working off of, a nerve cell is working off its ability to modulate ion movement. All right? In other words, allowing like charges, I should say unlike charges, to attract each other. And it's working off of a deliberate concentration gradient. It's created so that it can, in addition, make sure that this thing happens. So it's sort of important to make those kinds of real connections. So obviously understand what's going on here. In other words, to cause this spike. All right? Then, of course, the repolarization, the refractory period. All right? In other words, obviously understand what the refractory period is. Why? What causes it? Why does it happen? Why is it a good thing that it happens? All right? We know it's a good thing because it keeps the action potential moving in only one direction. That's what we want. All right? Also keep in mind that all nerve cells have a threshold potential that's built into the nature of the cell. All right? These were established many, many moons ago. All right? Different types of cells have different thresholds. For example, to just give you an idea, a practical idea of how this works. Embedded in your retina, you have two sensory receptors, nerves, sensory nerves. You have rods and cones. Okay? Well, as it turns out, cone cells, all right, in other words, to stimulate them, they need a higher amount of incoming light, all right, incident light. They won't turn on unless there's a certain number of photons literally being absorbed by the pigments. So, in other words, their threshold is different before they actually activate and allow you to see color. That's why at night, even if you're wearing all kinds of colors, we can't detect you as wearing all kinds of colors because your cone cells have not reached it, they're not being stimulated enough, they're not reaching their threshold. So the only cells that are operating in you when you walk out in the dark are your rod cells because they have a lower threshold before they fire. So they don't need as much light to stimulate them to get them to create these actions of attention. So they have a lower threshold. And that's all that means. So that's a practical example of what we're talking about. But all nerve cells do. And if you want to create an active potential, you will have to move enough charge in other words, within the axon to literally breach the threshold. And that's what this point is right here. And then suddenly, there's still enough pouring in. More does pour in at some point. Then you will